Good morning, my dear students. Welcome to our Zoology online class. Dear students, in the last class we studied about the genetic diseases. Okay, genetic diseases. The reasons I said, the reason for the genetic diseases: mutation in a single gene, damages or abnormalities in a chromosome, or abnormalities in a set of chromosomes. So these are the reasons for the genetic diseases. Okay, and the genetic diseases are classified into two major types. Okay, two major types. The first one is Mendelian disorder and the second one is chromosomal disorder. Okay, so if any one gene is affected or if any one gene is involved in the mutation, it leads to some diseases. That diseases are called Mendelian diseases. Okay, and then for the Mendelian diseases we have studied some examples. For example, the albinism, okay, phenylketonuria, thalassemia. Okay, so these are the examples for the Mendelian disorder or Mendelian disease. Okay, so last class we studied about this Mendelian disorder. So today we are going to study about the chromosomal disorders. Okay, so the second type of genetic disease. So there are two types of genetic disease. One is the Mendelian disorder and the second one is the chromosomal disorder. Okay, so today we are going to study about the chromosomal disorders. Okay, dear students, in this lesson, and many times I told you, our body cells consist of 46 chromosomes. Okay, all these 46 chromosomes are arranged in 23 pairs. Here the first 44 chromosomes are determining our somatic characters or body characters, so they are called autosomes. And the last two chromosomes, okay, that means the 23rd pair of chromosomes determining our sex, so they are called sex chromosomes, otherwise called allosomes. Okay, so the chromosomes which are determining our body characters are called autosomes, and the chromosomes which are determining our sex is called allosomes. Understand of you? Understand? Then, during the cell division, when the cells are dividing in their number, the chromosomes which are present inside the cells are also dividing in their number. Okay, it is a natural process. When the cell is dividing in their number, the number of chromosomes also dividing. This is a natural process. But because of some reasons, sometimes during the cell division, when the cell division takes place, okay, some chromosomes are not separated properly. Okay, some chromosomes are not separated properly. So if any errors occur in the cell division, the chromosomes are sometimes not separating properly. So sometimes the single cell, okay, the cells getting some additional chromosomes. The cells gain some additional chromosomes, or some cells lost some chromosomes. Okay, so when the cell division takes place, if the chromosomes are not separated properly, some cells getting some additional chromosomes and some cells losing some chromosomes. So this condition is called aneuploidy. This condition is called aneuploidy. This condition is called aneuploidy. So in this class, today we are going to study about some examples for these chromosomal disorders. And the chromosomes, already I told you, two types of chromosomes, autosomes and allosomes. So the chromosomes disorders also affecting both autosomes and also allosomes. Some autosomes are affected by some diseases and allosomes also affected by some diseases. So today we are going to study about this chromosomal disorder. So first we are going to study about autosomal abnormalities. Okay, autosomal abnormalities. Autosomes meaning you all know the chromosomes which are determining our body character or somatic character is called autosomes. Okay, dear students, listen very carefully. See, in your book there are two examples are mentioned for this autosomal abnormalities. The first example is a Down syndrome and the second example is Paget syndrome. Okay, so now we are going to study about two examples for this autosomal abnormalities. First, Down syndrome. This Down syndrome is otherwise called trisomy 21. Okay, Down syndrome is otherwise called trisomy 21. So the 21st chromosome is affected. Okay, so 21st chromosome is affected because of damage, or if some abnormalities or takes place in the 21st chromosome, it leads to one disorder. That disorder is called Down syndrome. That disorder is called Down syndrome. Understand of you? Okay. So the characters of this Down syndrome. Listen very carefully. See, if anyone's chromosomes, 21st chromosome is affected, we can see some important symptoms in their body. Okay, so the first symptom is they are poorly developed mental activities. Okay, poorly developed mental activities. Okay, so mentally affected. So this is the first character of these people. Okay, Down syndrome people. Then, second character is the central nervous system also not developed properly. Central nervous system means both the brain and spinal cord. So this nervous system is also not developed properly. And the third symptom is they have more space between the two eyes. Okay, more space between the two eyes. This is the third character of these people, Down syndrome affected people. Then the fourth one is their nose is very flat and the mouth is always opening, constantly opening, and the tongue is protruding outside. So these are some of the characters of the people, those who are affected with this Down syndrome. Those who are affected with this Down syndrome. So once I repeat the characters of these people. So the first I said, they are mentally affected. Okay, their mental development is very poor. Their central nervous system development is also very poor. More space is present between the two eyes. Then the nose is very flat. And mouth is always opening, constantly opening, and the tongue is protruding outside. So these are the characters of the affected people. Hope you understood. Then next one is Paget syndrome. Okay, it is otherwise called trisomy 13. Okay, Paget syndrome is otherwise called trisomy 13. So the 13th chromosome is here damaged, or some errors occur in the 13 chromosomes, and maximum for these chromosomal abnormalities are due to the errors in the or changes in the structure and number of chromosomes. Changes in the structure of chromosomes. Changes and the structure of chromosome is called what is the reason for the chromosomal abnormalities. Okay, then here Paget syndrome is always called trisomy 13. So first now I explain about the characters or the symptoms of these affected people, those who are affected with this Paget syndrome. Okay, see, their first character is they are also mentally poorly developed. Okay, their mental development is very poor. Okay, so it's the first character. And the second character is their head size is very small, but the size of the eyes are very large. Okay. So the size of the head is very small and the size of the eyes is very large. So this is second. Okay, and they are very tall. Okay, very tall and very big size. And the height is very high and the size also, and the size of the body also very high. So once again repeat, the first one is poorly developed mental activities. Then small head with large eyes. The second, then their height is very high. Okay, then cleft palate. Cleft palate meaning in this region a small burrow is present. In this region a small burrow is present. So this is the character of Paget syndrome. The people, those who are affected with this disorder. Okay. So hope you are understood. Then their internal organs also not developed properly and their functions also malfunctions. Okay, their internal organs are not developed properly and the functions of these internal organs also very low. Understand of you? So dear students, now we completed the two examples for the autosomal abnormalities. Then now we are going to study about the allosomal abnormalities. Allosomes meaning you all know the chromosomes which are controlling our sex or determining our sex is called allosomes. Okay, so in the sex chromosomes also some abnormalities are takes place. So example for this allosomal abnormalities is Klinefelter syndrome and Turner syndrome. Klinefelter syndrome and Turner syndrome. Listen very carefully, dear students. See, normally in the male body, one X chromosome and one Y chromosome. So two sex chromosomes are there. One is X chromosome and the second one is the Y chromosome. This is the normal sex chromosome in male body. But this syndrome, this disorder maximum affecting the male. Okay, the Klinefelter syndrome maximum affecting the male, and if any male is affected with this Klinefelter syndrome, in their body cells, 47 chromosomes are there instead of 46 chromosomes. Okay, listen very carefully. See, 44 autosomes. Okay, 44 autosomes plus one X chromosome and one Y chromosome. 44, 45, 46. So totally 46. This is a normal male. Okay, but the person who is affected with this Klinefelter syndrome, in their body, one more additional X chromosome is there. One more additional X chromosome is there. So in their body, totally 47 chromosomes are there. But actually 46 number only there, but here one additional X chromosome is added in their body cells, so they have 47 chromosomes. 47 chromosomes. Hope you understood the meaning of this Klinefelter syndrome. So Klinefelter syndrome is otherwise called XXY male, because this disorder maximum affecting the male, and the person affected with this disorder getting one more additional X chromosome, they have 47 chromosomes instead of 46. Okay, hope you all understood this Klinefelter syndrome. Then the symptoms or the characters of this Klinefelter syndrome. Okay, listen very carefully, friends. See, first one is they are sterile. These male unable to produce any gamete, any sex cells or any young ones. Okay, so they are the sterile male. Okay, they are also very tall. Their height is very tall and size is very obese. Okay, very short body. So these are the some of the characters. Then their limbs are very long. Okay, their limbs mean both hands and legs. Okay, that's why so the size of the limbs and size of the hands and legs are very large. Okay, then they have very poorly developed genital organ. Genital organs meaning one or reproductive organ. That means reproductive organs also not developed properly. Understand, my dear students? So these are the characters of this Klinefelter syndrome people are XXY male. Then second disorder is Turner syndrome. Second disorder is Turner syndrome. This Turner syndrome is otherwise called XO female, because this disorder maximum affecting the female. This disorder maximum affecting the female. Listen very carefully. So the female or the women affected with this disorder, this Turner syndrome, getting only 45 chromosomes instead of 46 chromosomes. See, so 44 autosomes. This is a normal autosomes. Then one X chromosome. They should have two X chromosomes. Okay, so 44, 45, 46. So 46. But here in these people, the women who are affected with this Turner syndromes getting only one X chromosome. Okay, so 44 and 40 plus 1 45. So they have only 45 chromosomes. One sex chromosome is absent in their body. So they will get only 45 chromosomes instead of 46 chromosomes. Understand, dear students? So I hope you all understood the difference between these two syndromes, Klinefelter syndrome and Turner syndrome. Then the characters of this Turner syndrome. The first main character of this Turner syndrome is they are sterile female, meaning they cannot produce the female gamete or female sex, or they cannot produce the young one. So sterile female, this is the major character of this Turner syndrome. Okay, then second one is they have webbed neck. That means the size of the neck is very short. Size of the neck is very short. And the size of the breast also very small. Okay, poorly developed breast. Okay, and they will not get normal menstrual cycle during puberty. They will not get normal menstrual cycle during puberty. So these are some of the characters of this Turner syndrome. Understand, my dear students? So dear students, now we have completed the second type of genetic disease, chromosomal disorder or chromosomal disease. Okay, and the reason for this chromosomal disorder is changes or errors in the number and structure of chromosomes. Okay, and this disorder affecting both autosomes and allosomes. And two examples are now we studied two examples for these autosomal abnormalities, and we studied two examples for the allosomal abnormalities. Understand, my dear students? So dear students, now we have completed this fourth chapter. Now we have completed the fourth chapter. Okay, dear students, now I am going to explain some important 2 mark questions of this chapter. Okay, so listen very carefully. Yes, students, in this lesson, many 5-mark questions, 3-mark questions, 2-mark questions, and many 1-mark questions are there. Whenever I take the class, I am telling you this is very important for 5-mark, this is very important for 3-mark, this is very important for 2-mark. Like that, I am telling. So now we are going to study about some important. I give some revision for this two-mark questions. Okay, some 2-mark questions. So listen very carefully. The first 2-mark question is multiple allele. Multiple allele. The first 2-mark question of this chapter is multiple allele. Multiple allele meaning 2 or more chromosomes. Sorry, 2 or more alleles. When 2 or more alleles located in the same location of a chromosome and controlling a single character is called multiple allele. Okay, 2 or more alleles are located in a single chromosome. Okay, the same locations of a chromosome and they are controlling a single character. These chromosomes, these alleles are called multiple alleles. So it's the first 2-mark question. Okay, then the second 2-mark question is kin cells. Okay, the second 2-mark question, important 2-mark question is kin cells. Kin cells meaning in honeybees. So this is one of the important phenomenon we can see in the body of the honeybees. Okay, actually in honeybee colony there are 3 types of honeybees are there: queen bee, worker bees, and drone bees. Here the queen bee and worker bees, both bees are developed from the fertilized egg, but the drone bees are developed from the unfertilized egg by parthenogenesis process. Already I explained these points. Okay, so here 2 female bees are there. The queen bee and worker bees both are female only in a honeybee colony. But here the queen bee only laying the egg. The worker bees will not lay any egg. Okay, but the worker bees indirectly supporting or indirectly increasing the reproductive capacity of the queen bees. So this phenomenon is called kin cells. This phenomenon is called kin cells. So in a honeybee colony, the worker bees also female, but they will not lay egg. They will support the queen bees to increase the number of egg or increase the reproductive capacity of the queen bees. So this phenomenon is called kin cells. Okay, so second important 2-mark question. Okay, then the third important 2-mark question is karyotyping. Karyotyping. Okay, so karyotyping is a technique. The help of this technique, we can separate all the 46 chromosomes in a cell from a cell, and we can arrange all these 46 chromosomes in a graph. This technique is called karyotyping technique. So karyotyping is a technique in which the complete set of chromosomes is separated and arranged in a graph. This technique is called karyotyping. Understand of you? Then fourth 2-mark question. The fourth 2-mark question is colon gene. Okay, colon gene. So when I explained this topic, I told you in each and every chromosome a particular number of genes are there. 46 chromosomes are there. In all the chromosomes a particular number of genes is there. And the genes are also present in the 6 chromosomes. Okay, genes are also present in the 6 chromosomes. Okay, here the genes present in different region of the X chromosome is called X-linked gene, and the genes present in different region of the Y chromosome is called Y-linked gene, otherwise called colon genes. Otherwise called colon genes. So the genes present in different region of the X chromosome is called X-linked genes, and the genes present in different region of the Y chromosome is called Y-linked genes, otherwise called colon genes. So dear students, now we completed 4 important 2-mark questions. Okay, and the fifth 2-mark question is applications of karyotyping or the uses of karyotyping. Okay, application of karyotyping. Say, there are 5 uses of in your book. There are 5 uses or applications of this karyotyping is explained. The first application or use of this karyotyping technique is this technique is used to identify the gender. When the baby is developing inside the uterus of the mother, we don't know what is whether this baby is male or female. But with the help of this karyotyping technique, we can identify the sex of the fetus. So this is the first application. And the second application is this technique, this karyotyping technique is used to find out the chromosomal aberrations. Chromosomal aberration meaning deletion, then multiplication, translocations. Okay, so like that some abnormalities we can see in the structure of the chromosome. This is called chromosomal aberrations. Understand of you? Understand? So these aberrations, chromosomal aberration, we can identify with the help of the karyotyping technique. Okay, so 2 applications we have studied. The third one is it is used to identify the aneuploidy. Aneuploidy meaning when the number of chromosome is increased or decreased in a chromosome in a cell due to the errors in the cell division. Okay, so if any errors takes place in the cell division, the number of chromosome is increased or decreased in a cell, this condition is called aneuploidy. So with the help of karyotyping technique, we can identify this aneuploidy. Okay, then the next one. This technique, this karyotyping technique is used to understand the evolutionary relationship between the species. This technique is used to understand the evolutionary relationship between the species. This is the 4th use. And the last one is this karyotyping technique is used to understand the genetic disease or used to identify the genetic disease. So once again I repeat all the 5 uses of this karyotyping technique. The first one is used to identify the gender or used to identify the sex of the fetus. And the second one is it is used to identify the chromosomal aberrations. Third, it is used to identify the chromosomal abnormalities like aneuploidy. Fourth, it is used to identify the evolutionary relationship between the species. And the last one, this technique, karyotyping technique, is used to identify the genetic diseases. Understand, my dear students? And the last question is pedigree analysis. Okay, here pedigree analysis meaning it is otherwise called family tree. Pedigree analysis is always called family tree. That means by using some genetic symbols, the scientist drawing some diagram to understand the characters which are appearing in a particular family for few generations, past generations. Okay, so pedigree analysis is used to understand the characters appearing in a particular family members from a past few generations. So this type of analysis is called pedigree analysis. Understand, my dear students? So dear students, today now we completed some important 2-mark questions of this 4th chapter. Okay, so today we have completed the chromosomal abnormalities and some important 2-mark questions we have studied today. Okay, dear students, so today's topic also very important topic, particularly this chromosomal abnormalities are very important for 5-mark question. Separately, it may be asked for 2-mark questions. Explain about Down syndrome or explain about Klinefelter syndrome. Like that, question may be asked. So, dear students, today is try to complete this topic, and we will meet in the next class. Thank you.